Okay, let's see that. Let's see the cell cycle phases. On that topics which from the biochemistry is the cellular one. Okay, let's see cell cycle phases. Okay, let's see that. Let's see that. We have four of that. We have G1, we have S, we have G2, and we have M. And then we have cytokinesis. Okay, for G1, it's the growth phase. It is the phase that before the DNA synthesis. And after the DNA synthesis, we have G2, and this is a phase after the DNA synthesis. And then we have mitosis. Okay, for a cell to give two daughter cells, it should bring DNA copy that's congruent to each daughter. So the DNA should be duplicated into two. Okay, duplicated means times two, and this it occurs in DNA synthesis. Okay, all of these parts of that cell cycle is called interphase. Okay, this is the phase in between mitosis, interphase. Okay, here in growth, you are increase the number of what? Of the proteins needed for the DNA synthesis, and you increase again and again organelles in the cells, and we will see each organelle individually in that video. Okay, for the DNA synthesis, we have this copy and this copy, it's duplicated, and then in G2 you're checking the mutation, or you're checking what happens in DNA synthesis. If there any problem with the DNA before mitosis, it will fix it. Okay, and we will see mitosis, and it has phases. Let's see the phases here. For mitosis or for M phase, this is the shortest phase of the cell cycle. Okay, it includes prophase, prometaphase, metaphase, anaphase, and telophase. Okay, for prophase, you will have the duplicated DNA here. You will have this DNA here. And this in metaphase, you will have them in lining, and this is a cell, and this is a cell. Then you will split them. This is called the ana, to split, and this is the phase, to split them. And we have a copy here, copy of the chromosome, one here, one here, one here, one here, and one for that, one for that, one for that. Okay, and this is telophase. Remember, telo it's apart. Remember, telo like telephone. So they are very distant. They will be apart from each other. Each chromosome or each copy of DNA, they underwent on the S phase. Okay, in duplication, remember that. This is very important. Okay, and you will have then cytokinesis. Here you split the DNA or the chromosomes or the genome, and then you will split the cytoplasm in the two cells, and this is called cytokinesis. It occurs after mitosis. Okay, let's talk about that and associate that with tumor suppressors, and associate that with diseases, and you may see that in your exam day of the board. Okay, here see we have checkpoints. We have checkpoints, control transition between phases of the cell cycle. Okay, this process is regulated by proteins, which is called cyclin. Remember, they are in, and they are proteins, and then remember they are cyclic, they are cyclins. Okay, to remember that, and to remember cyclin dependent kinases. Kinases, it will add phosphate group, and this is enzyme will add phosphate to the cyclin, and cyclin are dependent. They need kinases, okay, to be activated, so they need phosphate. And we have tumor suppressor. We will see the association between them just in 1 second. Okay, see here, we have a checkpoint here. We have a checkpoint here, okay, which is called G1S checkpoints. So to go to the DNA synthesis, to duplicate it, for preparation for mitosis, to prepare the cell to divide into two cells, you should pass the checkpoint, and you have restriction point to what? to restrict cell from multiplying like that in a crazy way. And to remember that medical term, this is a tumor. Okay, this is a crazy cell division, cell division like that, and this is a tumor. Okay, they are not functioning. Okay, they replicate out of proportion like that, like that, and then they press on the normal or healthy tissue. They they press on that. Okay, they take the nutrients from them, from the healthy working tissue. Okay, and when the body find that all of this cell division, it will undergo for these cells to decrease its number. So you are increasing the number of non-functioning cells and decreasing the number of the cells. It's a response from the body to down-regulate all of this uncontrolled cell division. Okay, and this is a principle behind the tumor. Okay, okay. Let's see that. We have G1. G1 is variable in duration. But remember, in phase. Remember, in phase is the shortest phase of cell cycle. Okay. Let's see the regulation of cell cycle. We have cyclin-dependent kinases, and we have cyclin-dependent kinases complexes, and we have tumor suppressor. Okay, let's see that. Cyclin-dependent kinases, they are constitutively expressed, but they are inactive when not bound to cyclin. Okay, okay. So you should have this complex to be activated, to activate this. Okay. The word constitutively expressed, it means that all of the time it's expressed. Okay. Here, remember, you have that gene, and you increase, you induce or express the protein. Remember that they are kinases, they are enzymes, so they are proteins. They are increasing in their number, constitutively. All this is crazy, but remember they are inactive. Okay, they are inactive. But when bound to cyclin, we will find this. We will find the cyclin with cyclin-dependent kinases complexes. Okay, so see that to activate them, cyclins are phase-specific regulatory protein. As I told you here, we will see their function right now. Okay, that activates cyclin-dependent kinase. Okay. These kinases are dependent on cyclin. Okay, it will activate cyclin-dependent kinases when stimulated by what? By growth factors. Remember here, growth factors. Insulin. Oh, very nice, bird in the video. Okay, insulin, and we have platelet-derived growth factor. We will see that on pathology. Okay, here with erythropoietin, we will see that on hematology. And here we have epidermal growth factor. Okay, all of these are growth factors. Okay, so cyclin that activates cyclin-dependent kinases when stimulated by growth factor. Here, the cyclin-dependent kinases complex can then phosphorylate. So kinase, they phosphorylate. Okay, they phosphorylate what? They phosphorylate other proteins. Here, like that. Here. This protein we will talk about in tumor suppressor, that's called retinoblastoma. This a protein that was discovered in a tumor in the retina, retinoblastoma, that was defective. So when the tumor suppressor was defective, here arising a tumor. Okay, in the retina, so it's called retinoblastoma. Okay, this is to know. Okay, this is not high, but just to know the naming of it. Okay, it will phosphorylate retinoblastoma to coordinate cell cycle progression. I don't understand this. Okay, I will show you in a minute. Okay, I will show you that. Okay, this complex must be activated or inactivated at appropriate times for cell cycle to progress. I don't understand this. Okay. See that. we have P53, which is called the policeman of the cell. Here is P53. When we find any DNA damage, like here, like here, if you have a damage in a gene, okay, you will have a damage in the DNA, okay. So you should undergo what? You should undergo apoptosis. Okay, okay. Kill that cell? No, it's not. It's apoptosis. It's the suicide of that cell. Okay, because it has a damaged DNA. So when you have a damaged DNA, it will induce P53. And P53 does what? P53 will induce a protein that's called P21. Okay, P53, it will cause P21 or P21 induction. So P21 will do what? It will inhibit cyclin-dependent kinases. So okay, they will inhibit cyclin-dependent kinases, and cyclin-dependent kinases does what? Or do what? Cyclin-dependent kinases, they phosphorylate retinoblastoma. Okay, what's the relation between retinoblastoma and between E2F? Okay, okay. For retinoblastoma, when it's hypophosphorylated, it is active, and when it's retinoblastoma hyperphosphorylated, it is inactive. So cyclin-dependent kinases, they phosphorylate retinoblastoma to make it inactive. Okay, and when it makes it inactive, you can progress from G1 to S, or the DNA synthesis, because E2F, it induces gene transcription that for proteins that will help in DNA synthesis and complete the cycle. So retinoblastoma attached to E2F to inhibit that, so it inhibits the progression of that. So when you have retinoblastoma in hypophosphorylated form, it will attach to E2F and it will inhibit gene transcription, and consequently it will inhibit what? It will inhibit the progression of the cell cycle. Okay, but when you have cyclin-dependent kinases inhibited, so retinoblastoma will be hypophosphorylated, so it will inhibit that. Okay, you understand me right now. Okay, you understand me right now. I think you understand me. Retinoblastoma to coordinate. Okay, to see that for activation G1S progression inhibition. Okay, so if we have mutation in tumor suppressor gene, it will result in what? Unrestrained cell division. It will undergo cell division, and you may find many tumor in different organs. Here, Lefromini syndrome. You may find different tumor, okay, in organ like leukemia, and you find here I will see Lefromini syndrome in detail in pathology. Okay, but I want you to remember here the mutation in P53, it will affect what? Okay, here Lefromini syndrome, loss of function of P53. So when P53 is lost, okay, you cannot induce B21. So you cannot inhibit cyclin-dependent kinases. So cyclin-dependent kinases will do what? Will hyperphosphorylate retinoblastoma, and it will be inactive. So when it will be inactive, you will have E2F, and it undergoes gene transcription, and when it undergoes gene transcription, it will increase what? It will increase protein needed for DNA synthesis, and it will go through the cycle. So you will have Lefromini syndrome. This is multiple tumor in the body, and you will find that in the case Lefromini syndrome, you will find multiple tumor in the family themselves. You will find the dad, you will find mommy, you will find my sister, you will find my uncle, all of them. Okay, they had the history of different cancer. Okay, and we will talk about that in details in pathology section. Okay, to remember, here we have growth factors: insulin, platelet, PDGF, EPO, EGF, all of that bind to a receptor that's called tyrosine kinase. Here tyrosine kinase. here we will see that on the cell, here is the cell, here is the cell membrane, and you will have a receptor. It's called tyrosine kinase. Tyrosine because here it has a tyrosine, which is an amino acid, and it undergoes autophosphorylation for its activation when growth factor attach here, so it will phosphorylate the tyrosine. That tyrosine, you will have the benzene ring and OH. Remember that from biochem. Remember that from amino acids. And if you don't remember that, don't worry. I will repeat that on amino acid section. Okay, for that tyrosine kinase receptor. And we will see tyrosine kinase receptors inhibitors like imatinib. Remember imatinib, remember CML. We will see that on hematology section. Okay, when growth factors bind tyrosine kinase receptors, it will do what? It will cause transition the cell from G1 to S phase like that, and we will complete the cell cycle. I think that cell cycle is clear right now. I think it's very clear right now. I think it's what is very clear right now. Okay, here we want to see the cell types from the point of view of what? From the point of view of what? Of the cell of G0 and G1, or the division. Okay, I don't understand this. Okay, I will explain that in a moment. In a moment. I think that USMLE is great. I think that USMLE is very, 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 very great. Okay, returning back. We have permanent cell type. We have a stable or quiescent. You know that they are old. We are old. This is a quiescent meaning. You know that my old grandpa, I want to go for the bathroom and undergoes. I have constipation problem with my motion. Do you know that? And we will have labile. Okay, there you go. Okay, they're creaky. Okay. Cell division again. Cell division. But it's not tumor. We will see that everyone has a cause. Here permanent, they remain in G0, but I know G1 and I know G2 and I know S phase, but what is G0? It will goes out from the cell cycle. I will not undergo cell cycle again. I'm too busy. I'm not going to have these children. I just work. You know that they're very important. They don't think in what? They don't think in marriage. They don't think in children. Just think in work. Like that from the neurons. Okay, the neurons. When you undergoes mitosis, you will stop your function during the mitosis. So, imagine that your brain stops its function. Okay, it can't happen. Okay, it can't happen. Okay, imagine that skeletal muscle. Imagine that cardiac muscle undergoes what? Undergoes mitosis. Okay, they will stop their function. And if you have that dog and you want to run, you will use all of that. To escape. But if they undergo cell division, you will be better. Okay, I think it will be bad. So they remain in the G0 to work all the time. Okay, to be alert. Okay, and you have RBCs. Why RBCs? Because mature RBCs does not contain nucleus, does not contain organelles or nucleus. They does not contain the DNA to undergoes the duplication. Okay, they are enucleated. They have removed the nucleus. Okay, so they are permanent. If you want to regenerate, you will regenerate from these cells that's that they don't have the program to be mature. They are immature, which is called the stem cells. Okay. For stable, for quiescent, enter J1 from G0 when stimulated. If you have hepatic injury, here this is the liver. If you have inflammation here, so you will regenerate that. And when you are from the quiescent, okay, you will return back to G1 to complete the cell cycle, to regenerate and restore the cells that were lost in that injury. or when you take a part from the liver in a liver transplant to give it to another patient, this donor liver will regenerate again. Okay, lymphocytes. Remember that lymphocytes have nucleus. Okay. PCT, proximal convoluted tubule. If you have injury in your kidney, you have, remember that nephron, and we will see the histology of nephron is very important. It's very important to see that on your exam. You have a glomeruli, okay, and you will have a proximal convoluted tubule, and then you will have the descending lobe of Henle, and then we will have the ascending lobe of Henle, and then you will have distal convoluted tubule, and then you will have collecting duct. If you don't understand this, if you don't memorize this, okay, we will see that on kidney, we will see that on kidney, or that urinary system. Okay, when it gets injured, it will regenerate from that quiescent or stable cell. They normally does not, okay, they normally don't undergo mitosis and enter the cell cycle, but when it's injured, you need to do what? You need to regenerate these cells. Okay, and we have the periosteal cells, the cells peri around that bone. You know that they are the outer lining of the bone. Okay, and when you will have injury again, you will need periosteal cells. Remember that, to regenerate periosteal cells. Okay, to regenerate the bone. Okay, and we will have labile. They never go to G0. They are too busy to divide. Unlike permanent cells, they get injured all of the time. Like what? Like gut epithelium. Oh, you eat, if you eat 3 times like people. No, we have friends that eat 10 times a day. Okay, so you will injure that gut epithelium. You will injure that intestine. You will injure that with this food. So it you know that it needs what? It needs to undergo cell division again to regenerate. Here, like the skin also, the skin get injured by friction and mechanical trauma to it. Okay, and the hair, okay, like hair follicles. And we have the bone marrow to do what? To produce the cells of the blood. Okay, RBCs, white blood cells, and platelets. You need RBCs and white blood cells and platelets. Okay, it will do what? You will have labile. So they undergo what? They undergo cell cycle. They divide rapidly, and these germ cells, they are the sperms and the ova. Okay, they are the sperms and the ova. They also, for females, they have what? They have the period every month. Okay, and for males, they have millions of sperms. Okay, and we will see that on reproductive. Labile are the most affected by chemotherapy. Okay, just not to memorize that. I just want you to understand the concept behind that. You know that chemotherapy, we give it to what? Or we give it to which disease? Which one? The cancer. Okay. Here, what's the idea between labile cells and cancer cells? What is the idea between both? They undergo cell division. For labile cell, they divide rapidly. So when you give chemotherapy, you are decreasing the cell division rate. So you will affect these labile cells. Beside that, you affect the what? You affect the tumor cells. So remember, people that take, it's very familiar that people that take chemotherapy, you will find they are bald, because the hair follicles, one of the cells that undergo or it's down the cell types of what? Of labile is hair follicle. Okay, so it affects hair follicle. Okay. For now, this is good. I want to tell you another thing. What is that? Here we have what's called human papillomavirus. We will see that in cervical cancer. Okay, we will see that in cervical cancer, and we will see that in skin. I will memorize you on that, but I want you to remember that human papillomavirus does what? It produces protein that's called E6 and a protein that's called E7. E6, it inhibit P53. So when you get infected with that virus and it inhibit P53, it will inhibit that, and it does what? Okay, okay, it does what? Oh, it does what? Okay, okay, it does what? Okay, to remember that. Okay, okay. See that? It will affect the P53, so it will undergo the cell division, cell division, cell division, cell division. But I want you to know again that